Good morning, everyone. Welcome to the fourth and final day of UMass Extension's Invasive Insect webinar series. My name is Tawny Simisky. I'm the Entomology Specialist with UMass Extension's Landscape, Nursery, and Urban Forestry program. And I'm excited to bring to you two more topics today brought to you by our program as well as UMass Extension's fruit program. Now, this series is made freely available to you through support by the Specialty Crop Block Grant Program at the U.S. Department of Agriculture. So we are very thankful to them for their grant support that allows us to provide these webinars. Before we introduce our two speakers for today, I do want to give a quick announcement about pesticide and association credits for those of you who are looking for those as a result of attending today's presentations. All instructions for receiving pesticide credit, including mass categories 25, 27, 29, 35, 36, 48, and the applicators or core license, as well as association credits, will be shared at the end of this webinar. So please make sure that if you're looking for those, you remain on until the very end to receive those instructions. It is also very important, especially if you are looking for pesticide credit, to answer all poll questions that are shared during these presentations. The answers to those poll questions will not be graded, but you must complete the poll questions in order to receive credit. If you sign off the webinar early or without having answered those questions, you will not be awarded pesticide credit. And then finally, a quick reminder about our gender, race, and ethnicity reporting. This is required in order to fulfill requirements as a recipient of federal funding. UMass Extension must report participant demographic information to the U.S. Department of Agriculture. This survey is 100% voluntary, so you can skip the entire survey or any question if you feel uncomfortable answering. This survey is also anonymous, so we are not collecting any identity or other information with this survey, and we will not be able to link your responses to participation in the program. So if you are willing to fill out this voluntary and anonymous demographic survey, you can find the link to do so in the chat on the control panel for GoToWebinar. So if you open up the chat section, you can find that link. Another place on the control panel that I should remind folks of is the questions area. If you expand that, you can send us questions that you have about today's presentations at any time, and after the speakers are done, we will get a chance to ask those questions of them and have them respond verbally. I also always forget to introduce Jeffrey Jouet, who is here helping with the technical support side of GoToWebinar, and I also have Ellen Weeks here as well, helping to answer some questions in the chat, so I thank them for their support during these invasive insect webinars. So without further ado, I am going to share my screen with our first speaker here, or request that he share his screen, and introduce to all of you Dr. Jaime Pinero. He is an Extension Associate Professor and a tree fruit entomologist with the UMass Stockbridge School of Agriculture, as well as the team leader of our fruit program with UMass Extension. And he will be talking about a 1-2-3 approach for spotted winged Drosophila and brown marmorated stinkbug integrated pest management in small farms. So please, Jaime, take it away, and thank you.
Yes, good morning, everyone. Thank you for joining this webinar. So today I'll be discussing integrated pest management options for 2 invasive pests that can be implemented in small farms. And I believe some of this information also applies to gardens. What I will do now is to stop the webcam so I can focus on my presentation. So back in 2013, when I was in Missouri, spotted winged Drosophila actually invaded the state, and that created a situation for growers. It was highly damaging. Missouri has a lot of small growers. So then we decided to use the concept of 1, 2, 3 IPM approach because to us, it was an easy way to explain and an easy way to understand by growers. So today, I will be talking about the 1, 2, 3 approach, which until about 2 years ago, I will say, for both species, on top you have a picture of the spotted wing Drosophila, and at the bottom is the brown marmorated Stinkbug. Basically, the 1, 2, 3 approach, it was monitoring, implementation of at least one cultural practice, and then insecticides. So for the spotted wing Drosophila, cultural practices include sanitation and canopy management. And for the brown marmorated Stinkbug, cultural practices, now we can talk about trap cropping. So what I will do today is I will focus on monitoring. I will not talk about cultural practices for the spotted wing Drosophila. I will briefly touch on insecticides because that will not be the focus today. Because now the focus is going to include biological control for both species. So that would be very good news. I think that there is an opportunity to implement biological control for each of these two insects. So let's go with the spotted wing Drosophila. Basically, this slide, the goal is to show you how can you construct a cheap trap for trapping spotted wing Drosophila. You can use it for monitoring. The only thing you need is a container which has small holes on the side, and those are going to be small enough to allow the spotted wing Drosophila to get inside the trap, but it will prevent larger insects from going inside. When you have too many large insects, then it's difficult to count these invasive species. So which bait can you use? Commercial lures are already available. You can go to the Grape IPM or the Alpha Sense websites, and you will find different brands and different companies, Sentry Lure, Trece, they produce lures. And those lures are based on fermentation materials. So keep in mind that no matter what lure or bait you are using, the number of flies captured are not predictors of the level of infestation. So this means that if you have blueberries, for example, and your crop is changing color, that means it's becoming susceptible to a spotted wing Drosophila infestation, and your traps catch few or several or many spotted wing Drosophila, you need to spray insecticides. Just because you are not catching spotted wing Drosophila or you catch a few, if the fruit is susceptible, you have to protect the fruit. So for the last 2 years, in my lab, we have been evaluating diluted Concord grape juice. You can see the proportions in the slide. Is 1 part of juice and 3 parts of water. For example, it could be 2 ounces of juice and 6 ounces of water in a trap. And we believe this is an effective, inexpensive, and easy way to get materials that you can use for monitoring. So we have found that diluted grape juice is about 3 times more effective to female spotted wing Drosophila than some commercial lures. And at the same time, diluted grape juice is also more selective because we're finding that attracts about 3 times fewer non-targets. And when it comes to costs, I mean, the cost of the bait, which is the juice, after diluting is about 9 cents per trap. So we have been doing work for more than 2 years. I will just show you one slide here, which is about what we did in 2020. So in 2020, in the early season, we compared the performance of diluted grape juice against 4 commercial lures. So the pictures have numbers, and those numbers match the type of lure that we tested. So number 2, for example, you can see the clear pouch is a Sentry lure, but all those lures, most of them are based mostly on fermentation materials. So the traps were deployed in late April, and they were inspected once a week. So what we found is that in one location, that was the place where we found the earliest spotted wing Drosophila captures. The first captures took place on May 25th. So 2 females were found in diluted grape juice baited traps, 2 females in one Trece trap, the number 4, which is more broad spectrum, and 3 females in the selective lure, also by Trece. And this graph shows the mean number of spotted wing Drosophila males and females combined across the 6 weeks that this test lasted. So as you can see, diluted grape juice performed quite well. And for this particular study, the Trece selective lure was the least effective compared to Sentry or the Alpha Sense or the Trece broad spectrum lure. At the bottom, you can see the cost of the different lures. Well, a few berry growers have asked me, okay, Jaime, for how many days can I test, can I have the trap baited with diluted grape juice, and for how many weeks it's going to be effective? So I didn't know because I have been testing only fresh material. But to answer this question, so we prepared diluted grape juice in the lab, and we age the grape juice for 1 week or for 2 weeks. Then we compared how attractive it was the 1 week old, the 2 week old juice versus fresh material. And here what you will see are the results. Basically, we found that fermentation influenced the response of females, but not males. So you have in blue the captures of males, and in orange the captures of females. And basically, only in females you see an effect of aging. So the 1 week old juice is more attractive to females than the fresh. When you go to 2 week old, it will be the same effect. There is no increase or decrease compared to the 1 week old. Here. And the proportion is about 2.4 times more captures in the 1 week old versus the fresh. And we already know that the fresh is more attractive in our studies than some commercial lures. So in summary, we have found that fermented grape juice is very attractive to spotted wing Drosophila, is more selective. And when you age the lure, basically you can have at least twice as many females compared to the fresh material. But I didn't show these results. But as you ferment diluted grape juice, it also becomes more attractive to non-targets. So then what I wanted to do is to be able to, if possible, minimize the captures of non-targets by adding borax or salt to the grape juice. And this is based on my previous work with the other fruit flies, where we are using borax to minimize fermentation. So basically, we compared fresh diluted grape juice, 1 week old with no salt. Then we have 1 week old that was aged in the presence of 1% salt. And we also have 1 week old juice aged in the presence of 2% salt. So here are the different treatments in the x-axis: fresh, 0% salt, 1% salt, 2% salt, and all these 3 treatments are aged for a week and then tested in the field. And in the y-axis, you will see the mean number of male and females captured per trap. So what we found, and this is similar to what we found before, is that when you don't have any salt and you age the grape juice, it becomes more attractive. So here you have not only females, but also males in this particular test responding in greater numbers when you age the material. But when you add salt, that was unexpected. You can have about twice as many coming compared to the age in the absence of salt, but it's about 3 to 4 times more than when you have fresh material. So salt seems to be helping with the spotted wing Drosophila captures. So now the answer to the growers will be, well, you could have your traps baited with the diluted grape juice. You can add 2% salt. I can calculate the amount, and it will be good for 2 weeks, perhaps more, because I never tested more than 2 weeks. But 2 weeks will be pretty good time period for collecting the flies and then count if you want to count them. So now let's go to biological control and the possibility of implementing. This is based on work conducted by the New York State IPM program. I believe it's very interesting what they have been doing in this regard, because the biological control agents that they are using actually are hummingbirds. You may have seen this information before, but basically new to me. I didn't know that. I went to online and I wanted to see how many species of hummingbird are in Massachusetts, but there's only one. Again, I didn't know that. But what is important to know here is that hummingbirds, they also feed on insects, and they can eat hundreds or thousands of insects every day. So what I would like to do now is describe some of the work that was done by Juliette Carroll and Grace Marshall with support from entomology and horticulture at Cornell University. So what they did is one study in Western New York. They had one raspberry farm and one blueberry farm. They were using hummingbird feeders at the density of 25 feeders per acre. In 2020, they compared these plots that have the feeders versus other blocks that have no feeders. And they were comparing captures of spotted wing Drosophila and also recording injury to the fruit. So the observations show that hummingbirds were present from June all the way to August in the berry plantings, and no hummingbirds, of course, that was expected. None of them were seen in plantings that didn't have any feeders. So again, they documented the injury and also captures of spotted wing Drosophila in traps. Every week, they were using traps baited with the sentry lures, and they also collected fruit to determine infestation. So the most important results is that in 8 out of 11 weeks, they found fewer spotted wing Drosophila in traps captured when you have the feeders compared to when there is no feeders. And when it comes to injury, they found the infestation was lower in those plots that had the hummingbird feeders. And in the box, you can see a statement by a grower that he believes that is a very good approach for using biological control involving hummingbirds. So I think what I would like to do this year, based on this work, is that I would like to be able to use maybe the traps, because I know the traps are going to be attracting spotted wing Drosophila. So next to the trap, I could have a feeder and then record or compare captures in the traps. And then what would you expect to find? So would you expect to find more spotted wing Drosophila captures in traps with the number 1 or in traps with the number 2? I would say I would expect to find more in the number 2. I have to say that these traps, or any trap, it doesn't capture every single spotted wing Drosophila. So I think it's going to be good in having a combination of the trap plus the feeder to be able to kill the ones that are not captured by the traps. So it's just a hypothesis. So again, when it comes to insecticides, I'm not going to present a list. There are many options available for some of them organic growers, of course, mostly for conventional. I only have one slide to tell you that when you use insecticides to control spotted wing Drosophila and you include sugar to the tank mix, that will help because sugar is a phagostimulant and it's going to make the flies be more exposed to the material, especially to the residue 1 or 2 days after they sprayed. So adding sugar is going to help, and this is based on research done by people in Connecticut and New Jersey. So let's discuss now spotted wing Drosophila. I have 50 minutes total. I already spent 20 with spotted wing Drosophila. So let's discuss the brown marmorated stinkbug. And I will just go directly to monitoring. So when do you need to spray against this invasive species? Well, using traps baited with the pheromone is the most important step you can take to determining when you need to spray. So thresholds, action thresholds have been developed for the sticky cards that you see on top and for the black pyramid traps that you see at the bottom in the picture. So, and these thresholds apply only to apple. And growers decide when to spray depending on the numbers. For example, in the clear sticky cards, when you have 4 cumulative captures per trap, it's time to spray insecticide. Again, I'm talking about apple. And when you have the black pyramid trap, when you have 10 cumulative captures per trap, it's time to spray. So where can you get these sticky cards? Where can you get these pyramid traps? There are different sources. One of them is the Great Lakes IPM. A different source is AgBio, and there are other sources of pheromone and traps. But what I would like to discuss now is the concept of trap cropping, which is a cultural management option for IPM for small-scale growers mostly. I don't think it's very feasible for large-scale operations, but trap cropping basically means that just like with children, I mean, if you give them the choice of having some ice cream versus a vegetable, well, chances are they're going to save the ice cream. So insects, they also respond in the same way. They have a preference for some plants over others. So trap cropping means that you are going to be using plants which are very attractive to the pest, and they are pulling these insects away from the cash crop. And this again can be implemented in small farms. And for the last, I would say, 10, 15 years, there has been a lot of research conducted on trap cropping around the country. And the target was mostly native stink bugs, but recently it has been more on the brown marmorated stink bug. Can you implement trap cropping as a way of managing this pest? So what you see in the picture is an evaluation done in Florida, where you have the cash crop is soybean, and in the picture you can see buckwheat in bloom, but next to buckwheat is a different trap crop. So they were comparing 4 or I think even more trap crops that included sorghum, sunflower, that included also early developing soybeans, buckwheat, and others. So based on this work, it has been suggested that sorghum and millet, they're pretty attractive to stink bugs, and those could be added to buckwheat and sunflower. So it depends on when do you need to do that. But I think it's more for the summer and through the fall. So what I wanted to do at UMass, it was to do a study evaluating trap cropping in combination with the pheromone. So the goal was to determine if we can bring more brown marmorated stink bugs to areas where you have trap cropping in combination with the pheromone compared to areas where you have the pheromone alone. So what is the contribution to trap cropping on the captures? And we evaluated 4 trap crop plants. The sunflower plants are basically dwarf and cultivar. They don't grow very tall. We evaluated sorghum, millet, and buckwheat. So we have 8 different plots where you have 4 trap crops in the presence of pheromone. Then you have the same 4 trap crops in the absence of pheromone. Then you have another plot, which is the pheromone alone, and then you have the unbaited control. All these plots receive sticky cards, 1 per plot, to capture this invasive insect. So here is a picture showing my graduate students working with me. I was taking the picture and preparing the plots. They were about 4 to 5 meters long and 20 square meters area for each one of them. And what you can see in this picture is on June 15th, you can see the trap crops already growing. By July 20th, buckwheat is already in bloom. And you can see the picture showing the sticky card with the pheromone at the center of the plot. Well, 2 of my graduate students prepared videos for my IPM class. And I want to show you this video because we have time and I think it's going to be informative. So it's going to be 1 video on the left, and then I will show you 1 on the right. It has some music, by the way. So basically, we found the brown marmorated coming to the trap crops, mostly when there is a pheromone, but I will show you the result in a moment. That's the pheromone lure with the sticky cards. It's called the ghost trap, but I will not be talking about that today. Those are the plots with the trap crops. Well, that was one video. The second one is this. It shows multiple adults walking on the sunflower. Also shows some injury based on pictures from the internet. And this is the one that Ajay wanted to include from his own garden on peppers. Again, the sticky card with the pheromone, the ghost trap that we have also evaluated. And this information comes from, originally was developed in Pennsylvania. So again, we are not saying that is ready to go. We want to do more evaluations. And I will show you the results. So the results are in this slide. Sticky cards having the pheromone lure in combination with trap crops. I combined the data across trap cropping species because there was no difference with sunflower or buckwheat, etc. So you can see that there were 47 brown marmorated adults captured in sticky cards when you have the pheromone on the trap crop, but you have about 40% fewer when you get the pheromone on grass in the absence of trap crops. But you only have one coming to the trap crops in the absence of the pheromone. And when you have an unbaited sticky card in an area which is not trap crops, we didn't get anything. So basically, we believe that there is an benefit of having a pheromone lure in combination with trap crops. And we're going to be testing this in more orchards this year. So now what I would like to do, again, is to discuss biological control options for the brown marmorated stinkbug. And this is very interesting because this wasp is called the samurai wasp, and it came to the US on its own around 2015, and currently is in about 12 different states. It's a very, very small wasp that the females lay the eggs on the eggs of the brown marmorated stinkbug, and the larvae of the wasp develops inside the egg. You can see in the picture on the right, the wasp coming out of the eggs after completing development. So this map shows first the status of the brown marmorated stinkbug around the country. You can see in red, those are areas or states where you have they have been reported as a severe damage to crops. In orange is a combination of agricultural and nuisance problems. In yellow is where there is just the nuisance problem only. And in green is where it has been found or intercepted, but it still has not been reported to be causing damage. So, but in the circles, you can see in yellow is the first report of the samurai wasp. And in gray, that will be the last report in 2019 of this wasp. So it's found mostly on the west and also in the east. Here is a video showing the life cycle of this wasp focusing on the eggs of the brown marmorated. It will be a couple of minutes long. I hope it's not going to be lagging. So as I was saying before, the females lay an egg inside the fruit. Inside the eggs of the brown marmorated. The larvae are going to ingest the contents of the eggs. They're going to kill the host. And then at some point, you have the adult wasps emerging from the eggs. Soon after that, they will mate with a male, and male and females will start looking for eggs where they can lay the eggs again. So this wasp has reported to be very effective in China and other Asian countries at killing eggs of the brown marmorated, to the point that with the help of other natural enemies, the brown marmorated that is not really a pest in China. So I think that would be enough. I will go to the next slide. So as I was saying before, the samurai wasp is able to attack up to 80% of the eggs in Asia, especially China. Here in North America, you have native wasps, which are parasitoids of native stinkbugs, but those are only able to attack about 10% of the brown marmorated stinkbug eggs. So because this wasp has been found in different states, now some researchers, they have been trying to redistribute this wasp in, for example, Cornell University, they have been releasing and monitoring in the state of New York since 2017. And Rutgers University, they have been releasing the wasps in New Jersey in 2018 and 19. I think Michigan is also now doing the same. So just take into account that when a female is killed by insecticides, for example, then we're preventing the successful biological control of the brown marmorated, because each female can lay more than 150 eggs. Here I have a very short video to show you how small this fly is and how is that they are kept in the laboratory. So the wasps are very small, as I was saying before. So again, I will not be talking about insecticides that you can use against the brown marmorated stink bug. For New England, you can go to the New England Tree Fruit Management Guide, which is free online, and that will provide you with the best options. Just keep in mind that if the wasp is present in orchards, well, they're likely to be killed by insecticides. So according to Oregon State University, the best place where you can release them, or for people doing research, it will be in unsprayed areas adjacent to the agricultural areas, but also in urban areas. So that's all what I wanted to discuss today. This is just one slide, because we have a seminar series in 2021, all online, and that's called the Mass Aggie Seminars. So we're going to be discussing multiple aspects of orchard trees in small scale production, and also pest management. So if you are interested, you can search for Mass Aggie Seminars 2021. And that's all what I wanted to say today. Thank you. You have any questions?
Excellent. Thank you, Jaime. We have lots of questions, so it's nice to have plenty of time for them. The first question is from Joseph. Can you please define what you mean by a small farm?
Well, there is the USDA definition, but I will not go with that because that's a farm that makes less than, I think it's $200,000. For me, a small farm is a farm that will be no more than 20 to 50 acres. It could be 1 acre to 5 acres. That's a difficult question to answer because this is based on my judgment. So a small farm, I would say it could also include family farms. But again, the US definition, the official definition, is a farm that makes, I think, no more than $200,000 per year.
Thank you. I'm also realizing that a reason why we have plenty of time is that we have to still run your poll questions. So can we maybe we'll do that now before we continue to have you answer questions from the audience. Is that okay?
Sure, thank you.
All right, Jeffrey, do you mind queuing up the poll questions?
Okay, the first question is up.
Excellent, thank you. So in New England, there are only 2 to 3 generations of spotted winged Drosophila because it reproduces very slowly. Is that true or false? And again, a friendly reminder, anyone looking for pesticide credit for attending today, please make sure you answer this as it shows up on your screen. Alternatively, if you're having trouble answering this question, through the prompt in GoToWebinar, you can put your answer in the questions area, and we'll get it that way as well. So we'll give folks a little bit to respond. I see we've got about 80% of folks answering, so if you can rush to get your answer in, again, especially if you need pesticide credit. And we also welcome those of you who don't need that credit to respond as well.
You have 10 seconds before the question closes. Okay, the question is closed. The results are 39% answered true, that 61% answered false. The correct answer is false. Want to go to the next question?
Yes, yeah, let's ask everybody the remaining questions for Dr. Pinero's talk.
The next question. One integrated pest management strategy that helps determine when SWD becomes active is.
Looks like folks are getting the hang of answering them now. We've got already about 82% folks responding, so keep getting your answers in. Great job, everyone.
Question closes in 10 seconds. Question closed. Results: cover crops 5%, monitoring 92%. So 92% got it right. Monitoring is the correct answer.
Great. Do we have one remaining question?
One more. Attract and kill of BSMB involves
Got about 80% of folks answering. Again, please get your responses in so we have more time for the ample number of questions we have from the audience.
So I'll close it in 10 seconds. Question closed. Okay. 42% answered spraying the perimeter trees. So that is the correct answer. A was the correct answer.
Well, great job on the poll questions, folks, and thank you. And we'll hop back on with Dr. Pinero here, and we'll continue to ask some of these questions. So another question, Jaime, if you're ready, from Michael. What
Yes.
kind of damage does the spotted wing Drosophila do to these crops? And then I will add to that from Jan, what crops are most affected or damaged by the spotted wing Drosophila? So can you talk a bit about all of that?
Yes, I omitted, apologize because I omitted those slides in order to have more time. I didn't know to what level people will know about this pest already. But basically, what happens with the spotted wing Drosophila is females, they have a serrated ovipositor. It's an egg-laying structure. So they insert the ovipositor inside the fruit. And this fruit is on the plants. It's sound fruit, it's ripening fruit. That makes a difference compared to other Drosophila species. So when the female lays the eggs inside the fruit, then you have one larva hatching and feeding inside the fruit. And the preferred host plants, it will be raspberry, blackberry, they love cherries, they go to blueberries, they go to strawberries. If you have peaches in your orchard and you let them get riper, well, when they get softer, they become more susceptible to spotted wing Drosophila. But I would say those fruits that have soft skin, so for example, they will not be able to lay eggs in apple. But grapes, to some extent, there is a slight likelihood to get damaged by spotted wing Drosophila. It's not very high, but basically all berries and some of the cherries and some tree fruits such as cherries. And the life cycle it happens very fast. In 3 or 4 weeks, you can have another generation. So you may have thousands of flies in just in a few weeks. So it's important to control them.
Thank you. Let's see, next question from Michael. What does the salt do to the fermentation process of the grape juice? Does it slow the fermentation process or in some way reduce the number of non-target species?
Well, that's a really good question. And I don't have the full answer because what we saw was the effect, but we are going to study the mechanism. So what I believe is that the salt is suppressing the growth of some bacteria, but the ones that survive, the ones that can reproduce in the presence of salt, maybe are adding some plant volatiles or some, not plants, some odor that is coming from the bacteria. So this is just a hypothesis because I have not tested this I have to do some analysis. But the answer is, I don't know what is doing to the fermentation, but we can tell you that it seems to be helping to attract more flies, especially females.
Thank you. Another question here from Joseph. If I spray every 7 days for spotted wing Drosophila, will that also take care of brown marmorated stink bug?
Well, I would say the brown marmorated can also go to berries, but they prefer more it's more arboreal. They prefer trees. So they would prefer to make, I think, to go to apple. And you don't have to spray against spotted wing Drosophila in apple. So I'm just thinking about the situation where you have a blueberry orchard, where you are spraying every week against spotted wing Drosophila. I would say it depends on what you spray, because when you spray a pyrethroid, it will be effective against brown marmorated Stinkbug and also against spotted wing Drosophila. But if you are spraying, for example, delegate, which is Spinetoram, then it's going to be effective against spotted wing Drosophila, but it will not be effective against the brown marmorated Stinkbug. So I would say it depends on what material is being used to control spotted wing Drosophila.
Thank you. Yes, I think more detail needed. Another question from Priscilla. I didn't understand the threshold for the sticky card. For what? 4 cards filled?
Well, the threshold will be if you have, for example, an apple orchard and you place the sticky card in the perimeter, as soon as you get 4 cumulative, so if today you got 2 and in a week you check again and you have 2 more, that will be total of 4. So it's cumulative per trap. When you get the 4, it's time to spray insecticides per sticky card.
Thank you. A question from Jan, and you might have answered this, but maybe just quick review. What crops are most affected or damaged by brown marmorated stink bug? Also, what type of damage do they cause and how extensive is it?
Well, I think the best example I can give you is in the mid-Atlantic in the year 2010. Growers were not expecting to have such high populations of this invasive pest. So I would say peach is one of the preferred fruits, apple, and the damage they cause is that when they feed, they insert the mouth part, which is like a syringe. It's a sucking mouth part, and as they feed, they deposit some saliva, which is toxic to the cells, the tissue inside the fruit, and this develops into corky areas. Sometimes growers don't see that until the fruit is in storage. But we have found in Massachusetts that in a few orchards, there has been about 1%, sometimes 2% injury. And that is because, again, when they feed, they deposit this toxic saliva, and this is going to cause some corky areas that developed inside the fruit. And of course, that fruit cannot be sold.
Thank you. Another question from Vincent. Have you attempted any studies for attracting the hummingbirds for spotted wing Drosophila using perennial planting strips with nectar plants they prefer? And he notes many of which would also attract other beneficial predatory insects and pollinators, which could be a separate study at the edges of crop fields, including some shrubs for nesting.
Well, yeah, basically, when I was reading this work done by Cornell University, then they have also a publication online. It's a blog, I think, and they talk about which plants can be planted to attract hummingbirds. So I read it, but I don't have all the information with me in my head. So I would refer you to that. You can search for Juliet Carroll, hummingbirds, and Cornell, and adding plants, because they report which plants are the most effective at attracting hummingbirds. But I was focusing more on the just the feeders. That's what I presented based on their work. But I am not very involved with the plants that can attract hummingbirds. I could discuss some plants that are really good at attracting pollinators or natural enemies. That will include buckwheat, and that will include sunflower, for example, and others. But I don't mean that those are going to attract hummingbirds. So my answer is, I don't know. I would refer to you to that publication by Cornell.
Thank you. A question from Bernard. Concerned about pollinators, do they or how many end up on the sticky traps?
Well, that's why we wanted to use sticky cards, which are clear. They have no color. And we know that when you use yellow sticky cards, you are going to be attracting some non-targets, including pollinators, including some parasitic wasps, and including some predatory insects. By using clear sticky cards, we're minimizing the captures of non-targets. And the pheromone only attracts stinkbugs. So it's not going to be the honeybees or wasps are coming to the pheromone. The pheromones are very specific to the insect you're trying to control.
Thank you. Next question from Pat. On the brown marmorated stink bug trap crop study, was there any contamination, quote unquote, from one crop to another? For example, would the results have been different if there had been a border of 5 meters or so between the plots?
Yeah, that's a really good point. We didn't have enough space available in the study area. That's why I didn't leave like a distance between plots. This year, we're going to be selecting only two trap crops, sunflower and buckwheat. And by having fewer trap crops, I can really include more distance between them. So the work that was presented, it was preliminary, but you can see a clear effect of the pheromone in combination with trap crop. What I didn't present is that when you have the trap crop species, the captures, when you have the pheromone, they were not very different. So there was some trend suggesting that sunflower was better than sorghum. But also what happens is that the plants don't grow at the same time. So you have buckwheat blooming very fast. In 4 weeks, the plants are in bloom. Sunflower, it takes longer. So that's why, for that reason, we selected only 2 plants for 2021 studies. But you're right. I mean, having a separation, it would have been ideal. We just didn't have the opportunity to do it. It will be done this year when we have just fewer treatments. It was too many plots. It was 10. Thank you.
Thank you. Another question here from Gary. Does the samurai wasp attack any insect eggs other than those of brown marmorated stinkbug?
I believe the short answer would be yes, but there are a number of researchers working on that. I don't have an update on to what extent they will affect native species, but they seem to have a preference for the brown marmorated stinkbug compared to native species. But to the extent to which they will attack natives, I am not 100% sure. I need to ask around.
Okay, you're doing great. Many more questions remaining. One from
No, and that's
Sarah.
why I wanted to have a shorter presentation and being able to answer questions.
This is great.
Yeah.
Thank you so much. From Sarah here, why did we see an increase in the brown marmorated stinkbug numbers in 2020 compared to previous years? Why are they coming into people's homes or houses?
Well, the numbers have been increasing gradually since 2000, I would say 8 or 9, 10. So before I came to UMass, I know there were people monitoring for these invasive species. The numbers were not so great, but they just continued to reproduce. 2019, there was a high number of insects also reported in homes. In some agricultural areas, that was the same case. But 2020, it was the year where we found brown marmorated in every single farm that we monitored, including in the western Massachusetts. So we included new farms in 2020, and every single one we found this insect to be present. Why they're coming to homes? Well, they seek shelter, they seek some warm areas, and they use pheromones to aggregate. So that would be one reason to why you see them around your house.
Thank you. Question from Jennifer. Are there plans to release the wasps in Massachusetts?
That is a great question, and we are planning to engage in those studies. The thing we need to do is we need to collect many brown marmorated from homes, which is not going to be easy, because those, if we can collect thousands of them or hundreds of them, then we can provide them with food, like a plants in the laboratory. So they have to be able to lay the eggs on those plants. Then we need to get these leaves and attach them to trees in different areas. Then we need to go back to these areas, let's say a few days later. We need to recover the eggs and then bring back the eggs to the lab to wait to see if this is called sentinel eggs. So you have them in the lab, you produce them, you bring them to the orchards or different areas, and then you come back to those areas to collect them and then determine if there is parasitic wasps. So it's not easy to do. We don't have a lot of people that can help with that project, but we are planning to do it because in New York, I mean, we know it's there, and they have been doing a great job at finding the wasp and then increasing numbers in the lab and then redistributing around the state. So that would be a long answer to your question.
Thank you. Another question from Richard. You mentioned the parasitic wasp arrived in the U.S., quote unquote, naturally. What does this mean as opposed to an introduced invasive pest?
Well, I would say that the introduced invasive pest in most cases is also accidentally. I mean, nobody plans to bring the invasives to the US. The wasp was found perhaps because it could be that in some of the brown marmorated that came in the egg stage, and the wasp was found, I mean, or present in those eggs. How it came to the US, nobody really knows how it was, how it made it to the US. But I will say the introduction is very similar. I mean, accidentally. There were people, researchers going to China. They were looking for the wasp. They brought the wasp back to the US. in secure areas, in laboratories, they were testing the non-target effects, and then it was found somewhere. I mean, it was not that escape from the lab.
Thank you. A question from Mary, excuse me, is grape juice better for trapping spotted wing Drosophila than cider vinegar with liquid soap?
I have not made the comparison. It could be tested, but if you ask me to guess, I would say chances are it's going to be more attractive. And I am not pushing to have people using the diluted grape juice. There is commercial lures which are effective. With the grape juice, is that it's just easy to get and cheaper, but it happens to be also more attractive. But more work needs to be done.
Thank you. A question from Priscilla. How prevalent is spotted wing Drosophila in Massachusetts?
Well, I would say, short answer, it's everywhere. So at this
Thank
moment,
you.
the adults are overwintering, and then you only need a few surviving the winter, and those few are enough to build populations. So I think you will find it everywhere.
Thank you. Another question from Ben. Do you have any tips for the management of brown marmorated stink bug in homes?
Well, I will say vacuum. Don't smash them because you know they release an odor, and they also are going to spoil the wall, and they're going to leave marks. So there is also homemade suggestions where you can have a pan with a soapy water. You can have a lamp, and this lamp is going to be like a LED light, and you can direct the light to the panel, and you will be killing a number of them if they're inside the house. Outside, I would just say washing them or vacuum.
Thank you. Maybe time for one more question from Priscilla. If we put out spotted wing Drosophila lures in April, in what month do we finish the monitoring season?
Well, monitoring, I think, doesn't have to start in April. I did it because I wanted to catch the very, very first insects, and we found them in mid-May. So I would say it's more related to your crop. I mean, if you have a blueberry plants, early season plants, I will go in mid-May. You can start monitoring. You are going to catch, I would say, not so many in the first few weeks, but as the fruits start ripening, I would say the highest populations are going to be found in August, late August. So when to stop? Since the traps are going to tell you only that the fly is present, and high numbers or low numbers are informative, I mean, that's good information. But since you have to protect the crop, I would say that when you are spraying insecticides, then monitoring becomes less relevant. So short answer, mid-May to, I would say, late August, but at that moment you need to be spraying insecticides to protect the crop. Trapping, monitoring is not enough to control flies. It's just, it has to be sprayed. You have to spray insecticide.
Well, thank you so much, Dr. Pinero. You've answered many great questions. There are many more that we're not going to have time to get to, and maybe if you're willing, I can send you those after the fact, and possibly we can post some responses on our website with the recording from today. Is to put you on the spot, is that okay?
That will be fine, no problem. Thank you.
Okay, great. Well,
Very
thank
good.
you so much. And I am going to take back control of the screen here and put up this little message. And now we have scheduled in our agenda for today a break, a 10-minute break, now a 9-minute break. So if everyone wants to stand up, move around, get yourself another cup of coffee, we will be back with our next presentation at 11:10. So thank you all very much, and please hang in there with us for our next presentation. Thank you again, Dr. Pinero. Excellent talk, and thanks for fielding so many questions. Okay, well, welcome back, everyone. It looks like it is 11:10, so I hope folks are back to their computer or whatever device you're watching the series on. And let me swap over and we'll get ready for our next presentation. Let's see, sorry about that. Multiple PowerPoints happening here. All right, well, again, thank you all for joining us today. It's so exciting. We've got over 253 folks joining us, and it's great again to have everyone back for these invasive insect webinars. And this next presentation is going to be a little bit different from our previous ones because we have multiple speakers that will be presenting information on a collaborative project that yours truly, as well as Dr. Pinero and others who I will introduce, are involved in. Now, our goal is spotted lanternfly monitoring in Massachusetts and New England, and to assist in testing airborne attractants for early detection. So, let me first introduce all of the speakers that you will hear from today. Again, myself, Tawny Simisky, Entomology Specialist with UMass Extension. Once I am done presenting some introductory or background information and a bit of a review about spotted lanternfly, I will hand things over to Dr. Miriam Cooperband with the U.S. Department of Agriculture, and she will talk about an overview of her trapping and lure and pheromone research that applies to this project. Next, we will introduce Dr. Jaime Andersen, who I believe will speak on behalf of Dr. Joseph Elkinton and their lab at UMass Amherst in the Department of Environmental Conservation. He will cover an overview of our integrated research and extension project that is funded through the Center for Agriculture, Food, and the Environment at the University of Massachusetts Amherst. And then Dr. Jaime Pinero will hop back in and close out our presentation and discuss the risks posed by spotted lanternfly to fruit crops, as well as supporting the mass monitoring effort. So, without further ado, let me hop into my portion of today's presentation and just start off with a little bit of a review about the pest itself, spotted lanternfly, Lycorma delicatula. Of course, this insect is sometimes commonly referred to as a lantern moth. It is neither a fly nor a moth. It belongs to the order Hemiptera. So these are the true bugs, the cicadas, hoppers, aphids, and other insects with piercing sucking mouth parts. So they use these piercing sucking mouth parts, as Jaime I think described, something like a syringe to remove plant fluids. So in the case of the spotted lanternfly, it is capable of feeding on many different plant parts for hosts that I will discuss in a moment, and remove those plant fluids. And then a byproduct of this is that, of course, like many of our Hemiptera, they produce a liquid and very sugary excrement known as honeydew. And this excrement can cause annoying situations, if you will, where it can coat leaves, it can coat parked cars that are beneath infested trees or patios, any outdoor furniture. And then on top of that sugary liquid surface, you get the development of sooty mold, which grows on top of that honeydew. That sugary excrement can also be very attractive to stinging insect pests, and so that can cause trouble as well, unfortunately. The spotted lanternfly is a member of the family Fulgoridae. These are also known as the plant hoppers, and so we know this insect can hop or jump. And then adults, of course, have wings, and they can then use those wings in combination with that hop to help them fly. I know that in previous webinars in this series, everyone has seen this map provided by the New York State Integrated Pest Management Program, but I did want to show this as a review again in case we have new attendees here who haven't seen it yet. And mention, of course, we know spotted lanternfly was first detected in 2014 in southeastern Pennsylvania, and we can see these areas that are highlighted in blue on this map that indicate an active spotted lanternfly infestation. So those are areas where we know that spotted lanternfly is present in the environment in those locations, is reproducing, and is a problem, unfortunately. The areas on this map that are shown in these sort of pink little dots, and there were some here, one present in Massachusetts as well, those represent individual finds of the spotted lanternfly where no established infestation is present. So for example, in Massachusetts, we had some multiple reports about single dead individual spotted lanternflies that were detected. And this is indicative of the fact that this insect is great at hitching a ride and unfortunately can move around with our unintended assistance quite readily, and we'll talk about that in a little bit as well. The Mass Department of Agricultural Resources stated previously in a webinar in this series that they have had these single detections in Billerica, Boston, Concord, Milford, Norwood, and Sharon, Massachusetts. Again, those are dead individuals that were detected but do not represent an established population of spotted lanternfly in our state. So we are, of course, hoping to avoid that. All right, so I will take care of the poll questions here for our presentation through my portion of this talk. So our first poll question, and have a look here, is which of the following best describes your knowledge, your own knowledge of the life cycle and identification of spotted lanternfly? So do you consider yourself an expert? Could you probably identify it if you had to? Do you need a refresher? And or do you fall in the category of what is the spotted lanternfly? So again, please answer this poll question, particularly if you are looking for pesticide credit. And this is great. I can see 80% of folks voting, so keep getting those answers in. Of course, there's no right answer to this question, but it will help me to figure out how fast or slow to go through the rest of my part of this presentation.
Can you see the results, Tony?
Let's see, I can see it here. 50% or 57% of you said you probably could ID it. 9% want to know what the insect is. 20% need a refresher. Some 14% are experts. Excellent. So let me say that I'll probably go moderately fast through the rest of this. But to give that bit of a refresher and to let us all remind ourselves why we are concerned about this insect, a recent publication, I believe at the end of 2020, has said that host plants for the spotted lanternfly now include at least 103 different species, and of course, if not more. And I've selected a few that, of course, are going to be significant for those of us working in managed landscapes. Again, me coming from the Landscape Nursery and Urban Forestry program at UMass Extension. One I've highlighted here, I think most of us know, the tree of heaven. There is this association with this insect and the tree of heaven. Of course, the tree of heaven is a non-native invasive plant itself, something that is not desirable and that we don't really want in our landscapes either. And unfortunately, spotted lanternfly is not restricted to feeding just on tree of heaven, and we can see the many different species of plants that it impacts here, including managed crops. So apple, plum, cherry, peach, apricot, grape. We know that there has been a significant impact to grape in Pennsylvania, and perhaps Dr. Pinero might mention that a little bit as well. So we know that there are crops and trees and shrubs that we care about that will be impacted by the feeding of this invasive pest. Having Tree of Heaven nearby is not a requirement to have spotted lanternfly in a location, but of course, because they do show a preference for that plant, it doesn't help. Let's see the next slide here. So I do want to give a quick review of the identification of this insect as well as its life cycle. The notes that I have on these and subsequent slides regarding the life cycle of this insect do refer to timing for what happens during the life cycle of spotted lanternfly in Pennsylvania. So while we expect that the timing could be very similar, were it to become established in Massachusetts, it might not exactly represent what would happen in our state. We know that adult females will lay their eggs in rows, and I'll show an uncovered egg mass momentarily here, but once she is done laying her eggs, she covers that with this waxy secretion that is white when it is first laid, such as pictured here. There are 30 to 50 eggs per mass, and at least 2 masses are laid per female. That secretion that she covers the eggs with, that dries and cracks and darkens and looks more like dried mud over time. It may not cover the entire egg mass, and through the process of aging and weathering, again, can get worn off. Egg masses are roughly 1 inch to 1.5 inches long and 1/2 to 3/4 of an inch wide. And so in Pennsylvania, again, eggs are seen beginning in late September or October and seen through June or following egg hatch. And I always like to show this great photo from my good friend Greg Hoover, formerly of Penn State. Spotted lanternfly egg masses compared to our very familiar, unfortunately, here in Massachusetts, gypsy moth egg mass. So that gives you an idea relative to size and color. And I always like to remind folks that aren't familiar with spotted lanternfly, but are familiar with gypsy moth, don't accidentally confuse the egg masses of spotted lanternfly for, say, a funky gypsy moth egg mass. It's not a gypsy moth female having a bad day. It's the egg mass of a separate insect. So nymphs will hatch from these eggs beginning in late April to early May. Again, the timing there for Pennsylvania, and we can see these teeny tiny insects that are beginning to emerge out of these eggs. This picture here, and hopefully everybody can see my pointer on the right, we can see the eggs themselves laid in rows here, and this has been an egg mass that is not covered, or that coating from the female has worn away. There are 4 nymphal instars, meaning there are 4 stages of juvenile or immature insects for the spotted lanternfly. So the first 3 of those are black with white spots, and they are present roughly from May through the end of June. So we can see what those instars 1 through 3 look like here. The 4th instar, so the most mature immature, if you will, the oldest of that immature stage, they typically are present the first week of July and add that red coloration in addition to the black and white spots. And we can see, again, this is an immature spotted lanternfly. We can see these wing buds pictured here, so that we know only the adults of this insect, as with many of our insects, have fully developed wings. And here are the adults. We've got, again, these are photos from Pennsylvania, courtesy of Vivian Dutton. We've got an image here of a tree that's absolutely covered with clusters of these adult lanternflies, and I've zoomed in on this one here to show adults typically present by mid-July and live until the first couple of hard frosts get them in Pennsylvania, and the timing for that may be roughly November to December. So second poll question, Jeffrey, would be which spotted lanternfly life stage is pictured here. So everybody have a look at this picture for a minute before we run that poll question. But is this an egg mass, a first through third instar nymph, a fourth instar nymph, or an adult spotted lanternfly? I can see around 85% of folks are voting. So if folks can keep voting, this is great. And you're all doing excellent. I'd say you are all almost experts here. And definitely keep getting those answers in, especially if you would like to receive pesticide credit for watching today's broadcast.
Tawny, I don't know if everybody can see the picture that the poll question refers to.
Yes, unfortunately, it disappears when we have to run the poll. So we gave a few seconds in the beginning to allow for that. And then unfortunately, it does disappear. Are we getting folks in the chat saying they need to see it again? Let's see. I can sort of try to, should I try to verbally describe? That might be bad. Oh, here we go. Jeffrey is opening that up again so we can see the image here. Jeffrey, are folks going to be able to answer again, or do we have to give the results?
They cannot
Okay.
answer. They cannot answer again. So,
Okay,
but
so
most of them are answered anyway.
okay, I did want to say for anyone who was unable to answer and does want pesticide credit, just please enter your answer right now before we, well, you can see the answers, I guess, but enter your answer into the questions section, especially if you want pesticide credit. So I can see here that 88% of you got this correct. This is a fourth instar nymph, and again, we can tell because the latest instar gets that red coloration that develops. All right, so we can close out this question and I will continue. Here we go. Oh, okay, so I did want to, at the risk of my internet failing me, try to show a quick video here, again from an orchard in Pennsylvania, and hopefully this will play, but again, this gives you another idea of why we do not want spotted lanternfly anywhere. Certainly we don't want it to come to Massachusetts, and we don't want it where it is now, but you can see many of the adult insects climbing in this tree that I apologize is oriented horizontally when it should be vertically, but we can see all of the adults climbing on this tree in this orchard, and the population is so high to the extent that one has landed on the camera. So that gives you an idea of what these other states are dealing with. I did want to just end my part of this presentation with a note about impact. The adults, as we've seen here, can be present in very high populations. This can be disruptive to residents and impact their daily life. Let's see here, sorry about that, my slides were delayed. The quality of life will be changed for many, and this is language from the Pennsylvania Department of Agriculture. They know what they've been dealing with and have been very vocal about sharing information with other states and trying to increase awareness so that we can avoid sharing in this problem and having this pest. And we can see here this tree absolutely coated with adult lanternflies. There's kids' toys pictured in the background, and they have another image here of what the sooty mold can look like as a result of, again, that honeydew dropping onto plants beneath where these insects are feeding, and that sooty mold developing on top of that honeydew. Again, I mentioned previously, stinging insects can also be attracted to that, and they can be a nuisance. In terms of trees and shrubs, more information is being gathered about that. I think the overall longer term impacts to trees and shrubs might yet be unknown. However, folks at Penn State and other researchers have noted that in certain tree species, they're seeing flagging of branches and maybe a little bit of dieback as a result of extensive feeding from the spotted lanternfly. And certainly out of Pennsylvania, they have reported an increase in labor costs for nurseries to meet state quarantine requirements for this insect. So that is another impact to think about as well. And so our last poll question here, I do want to wrap up so that we can move on to our next presenter in this presentation. But again, a situation here where we need to have a look at this photo before we run this. So everybody have a look here. How many spotted lanternfly egg masses do you see in this photo? So try to count up the visible egg masses that you can see in this photo very quickly. Maybe give a couple more seconds and then we'll run the poll question so that you can respond.
Start enough time.
Let's see if people are saying anything in the chat. I think that's probably good, Jeffrey. We'll give folks a chance to respond now. And that's great. Again, I can see some folks sending their answer to the questions area. If you're having trouble responding in the poll question function, you can send your response into the questions area.
Okay, 92% of vote, and I'm gonna close the question.
Thank you, Jeffrey. Okay, let's see. Oh, and I'm not sure if my screen is still sharing here. Let me try that again. Huh, I am wondering why it's not showing. Sorry about this, everyone. Try this again. Show screen. There we go. Okay, so I've circled this for you. What I could easily see was 9 egg masses in this photo, and you can see all of them circled here. Although, and if you can see my pointer, there might be a couple hidden back here, but I think my main point with this is that to remind everyone, while these insects do and can lay their egg masses on susceptible hosts, they can basically, like gypsy moth, lay their eggs on any flat surface. And so I believe this is the stair portion of a slide that was outside. Again, this courtesy of the Pennsylvania Department of Agriculture. And so you can see if you do some math based on 9 egg masses and 30 to 50 eggs per mass, if you were to move this slide to a new location, you could potentially be moving 270 to 450 spotted lanternflies to a new spot. So highlighting again the responsibility that we all have to be careful and vigilant to make sure that we're not accidentally moving this insect to new locations. All right, so let's see. And lastly, the last point that I want to make and item to share here, and I'll put this in the chat the next chance I get, but if you are living and working in Massachusetts, a friendly reminder, If you suspect that you've seen any of these life stages of the spotted lanternfly, please take a photo, collect a specimen, absolutely, if you can safely do so, and note your location, and then report it online here. And again, I'll share this link in the chat momentarily. If you are living and working in any other state outside of Massachusetts, you can also contact your state Department of Agriculture or your local university extension and report any suspicious insects to them as well. Okay, so with that, I am going to introduce our next speaker. And Dr. Cooperband, I'm going to try to give you control so that you can show your screen in a moment here. And sorry about my delay, I've got screens and things all over the place here. But our next speaker is, of course, Dr. Miriam Cooperband, again, working with the United States Department of Agriculture, and she will be talking about her research on spotted lanternfly attractants for development of lures. So please take it away. Thank you.
Okay, can everyone see my screen?
Yep.
Yes,
okay.
Perfect.
So I will just start by introducing some of the folks who are working on this project with me. We've been working on developing attractants for spotted lanternfly since just about when they arrived and were discovered in Pennsylvania. We started in 2015. And we started with looking for caromones, we host plant volatiles that they're attracted to. And we did come up with a lure, the methyl salicylate lure, which does help attract them in low density situations, but they aggregate and it's very difficult to draw them away from those aggregations. So when the densities are higher, that lure is less effective. So we're looking at developing lures using SLF volatiles and odors that SLF produce themselves, potentially a pheromone. We don't know if a pheromone exists, but we have evidence for it. They could also be using other things like sound. However, we've been working on this and we have some promising results. So this is me and my postdoc, Hud Jarfal, who has done a lot of the work that I'm going to talk about. And my two technicians, Isaiah and Sam. Isaiah did most of the bioassays in the lab. And then this is Kelly Merman. She's the lead on the field studies, and her crew who help in a number of different projects involving SLF, like developing traps and radio telemetry of SLF and learning about their biology. So you saw that there are 4 immature instars and then there are the adults, but it's not so simple because the adults are very long-lived. They arrive in late July, they emerge in late July, and then they live until late fall. So they typically die after oviposition is done around the beginning of November or when there's a hard frost. So what are they doing during that time? Well, I've divided up the stages into clearly differentiable stages. And the early stage, I divided it into 2 sections. And it's really focused on intensive feeding, but at the second half, their sex ratio changes and males become really hard to find. We don't know exactly where they're going, but right before we start to see mating, they come back. And then this happens between when they first emerge in the end of July and middle of early September. Around mid-September, we start to see mating, and I call this timing mid because when they start to mate, they're in a different physiological state. And we also see these massive flight events where they seem to deplete their resources because they're feeding in such great numbers that they have to move to find food. So they take off in clouds and fly quite far. And then in October or around the end of September, these dates are variable based on the year, and also the location. But they start to oviposit, and I call that stage late. So what do I mean by intensive feeding? I'm going to show you some examples. This is a single tree over time. In 2018, we were monitoring this tree, and right when the adults emerge, you can see the tree still has honeydew on it from the feeding. This is an ailanthus tree from feeding of nymphs, but it's normal. You see some adults on it. But as time goes on, and this is a sticky band, a bug barrier band that we were monitoring it, this white stuff is honeydew building up, the sugary excrement, and it's accumulating. And then the black stuff is sooty mold. And as you can see, the next week or two later, it's even more. And you can see the aggregations forming, and it's just completely coating this tree. We call this jokingly pudding because it's just thick and gooey and covers the whole tree. And this is a zoom out of the same tree. It's just completely covered. And so that is intensive feeding, and it does have an effect on the trees. And then by the end of a summer, right before mating time, this tree might not be such a good source of food anymore because they've really sucked it dry. So they will take off and fly and find new places. And unfortunately, that's part of what drives their spread. So they build up on Tree of Heaven during the adult stage. And right before mating, we really see the majority of the adults are on Tree of Heaven. And when they're ready to mate and right after mating, the female abdomens swell considerably. You start to see the yellow between the segments, and their eggs are developing. So the question is, if we're going to try to find a strong, powerful attractant, we want to know who is attracted to whom and when for mating. So to do this, with these stages in mind, we extracted females and males of different stages by dipping them or washing them in solvent. And then that solvent was used in bioassays. This is a Y plate bioassay where we have a control on one side and the solvent odor on another side, and air is moving into the plate through these tubes, and air comes down through this chamber, and we release an adult, and it can make a choice. And this is called a Y-tube bioassay. And so we tested the extracts from males and females, 2 males and females of the different stages. And what resulted is this. So here we have the living adult that was tested, and the males up here, females here, and the stages that they were in. They were field collected adults. And here we have the body extracts that were tested, male volatiles and female volatiles from the different timing from the field. And males were not attracted to the odors from males. Females were not attracted to the odors from females. There was a little attraction here with early 1 females to early 1 males, but it disappeared. But males of early 1 and early 2 and a little bit of mid were attracted to early 1 and early 2 females. And once mating started, there was no more attraction. So this looks like there could be evidence of a sex pheromone because males were attracted to females and this attraction stopped once mating started. So how do we use this? We need to find out which compounds in those extracts were responsible for the attraction. So one tool we have is called electroantennographic detection, where you hook up the antenna of the insect to electrodes and you run the extract through a gas chromatograph, which separates the compounds in time, and then they come out, spread out one at a time over, and they come out onto the antenna. And the antenna, when it detects something, it sends an electrical signal to the brain, and we pick that up with the electrodes. So we can see as the compounds are coming out which which are antennally active, which ones the insect can smell. However, the problem with spotted lanternfly, and this is the problem with the host plant volatiles as well, is that its antennae are able to detect almost everything we send over them. So it doesn't narrow it down quite enough. So what we did then was we compared the chemical profiles from the males to those from the females, and we looked for differences. And when we identified compounds that were different from males and females, we developed a synthetic blend of those compounds. And then we tested the blend in the Y tube, and what we got was attraction of males. So we were on the right track. So the next step would be to field test, because what you find for behavior in the lab doesn't always translate to the field perfectly. It often needs tweaking. Sometimes you don't have all the compounds, or sometimes you don't have the right ratio or dose. So you need to bring it to the field and test it for efficacy. So in 2021, this coming summer, we're planning to test this blend in the field for both efficacy and to determine a dose that's the appropriate dose to use. And the blend will be formulated into a lure that is an experimental matrix that we can apply to the tree trunks in different amounts. And this is the trap that was developed by my colleague at APHIS, Joe Frances, and it's called a circle trunk trap. It's similar to the pecan weevil trap, except at the top there's a big bag where we can accumulate. These bags will fill up completely with spotted lanternflies. And this trap takes advantage of the movement and behavior of the insect, because spotted lanternfly typically climb up tree trunks. They love to climb up, and they will fall out of the canopy and then they'll climb back up again. They're not strong flyers. In addition, it excludes a lot of bycatch. The sticky traps and the sticky bands, a lot of them catch vertebrates or a lot of other insect bycatch. This one is pretty specific to spotted lanternfly, especially when placed on ailanthus, which doesn't have a lot of insects feeding on it. So the plan is to test this new blend at 0, 10, and 100 times dose to see if we can figure out approximately what the dosage should be, and hopefully we'll see some attraction. And in doing this, we're going to be collaborating with UMass Amherst and several others in Pennsylvania and other states. So, and with that, I'm going to hand it off to one of our collaborators at UMass Amherst.
Great, thank you. And I will invite our next speaker, Jaime, to share his screen and can introduce, whoops, sorry again, Jaime again, Dr. Andersen, working with Dr. Joseph Elkinton's lab at UMass Amherst in the Department of Environmental Conservation. So Jaime, please take it away. Thank you.
All right. Well, Tawny, thank you so much. And building off of what Dr. Cooperband just presented with the testing for attractants, I'm going to share with you an integrated research and extension project that we have, which is funded by the Center for Agriculture, Food, and the Environment here at UMass, to try to work with stakeholders, hopefully you in the audience, to test these attractants that Dr. Cooperband is developing and some other methods as well. I'm going to shut my camera off and we'll dive in. So as Tawny mentioned earlier, spotted lanternfly is, thankfully not established yet in Massachusetts, but we do have reports. And if you saw, especially in October, there was several news articles. There was the spottings outside of Boston and Billerica, and we're really interested in like how can we work with stakeholders to identify these populations before they get established. And as we've mentioned already, spotted lanternfly has an incredible host range. On the left here, we have a range or a sample of both native and ornamental plants that spotted lanternfly can survive on and will use at different life stages. As Tawny showed the photos of the different instars, you can see that different species, the instars have different abilities to persist and different attractiveness. They really like Tree of Heaven, but they can also do very well on hops, on butternut, on black walnuts. And these are all trees that we have kind of native here in Massachusetts, or at least are widespread. And on the right, this is a climate suitability map. If you see the red coloration, that means highly suitable habitat. Orange is moderate suitability, and green is low suitability. White would be no match. But all of Massachusetts essentially has some type of suitability, with from the Pioneer Valley to the east being either high or moderate suitability. So this is definitely something that we're concerned about establishing and having impacts both for growers, for homeowners, for nurseries. As Tawny mentioned earlier, it's really a big cost burden for Pennsylvania nurseries right now to go through their stocks in order to prevent the spread. So that's something we're hoping that we can have a project established so that it's not necessary here in Massachusetts. And what we're going to do is, as part of this project, is work with a variety of stakeholders. We're going to work with fruit growers, particularly apple and grape growers, and Dr. Pinero will talk about that in a few moments. We'd like to work with tree farms and nursery managers and other green industry professionals, as well as homeowners. That's one reason I'm excited to give the talk, is to just kind of get the information out there. So if you're interested, you can contact us and we can hopefully work with you as part of this project. And really, I think this project is a great way to visualize the importance of citizen scientists, not just for this project, but in general. But citizen scientists have played a really important role in SLF detection. This is a map on the left of reports from a database called iNaturalist. This is where people using their smartphones can take photos or traditional cameras and then upload them and share them. And these are, you can see the two records right here in Massachusetts. Another reason that we think that SLF will become established, if it's not already, and widespread, is that Tree of Heaven, which is the right-hand photo, is really quite widespread in Massachusetts, at least, Connecticut and Rhode Island. So there's suitable habitat, there's alternative hosts, and we have a suitable climate match. Our approach for this project is kind of like the best defense is a good offense. So we're hoping that we can work with these lures that Dr. Cooperband just presented, these novel airborne attractants, and compare them with methyl salicylate, which she presented as well. And one reason that we want to compare the two is that in Massachusetts, we will be working in a low density population if they're established. So maybe these two attractants have different success rates in high versus low densities, and that's what we want to look at. And because we're interested in getting a variety of stakeholders involved, we also want to look at the difference between using these aerial attractants to something that might be a little bit easier for a homeowner to use, such as a tree band, a bug barrier, or tanglefoot. And part of the UMass Extension Invasive Insect Seminar Series, you may have seen a presentation from Dr. Julie Lockwood at Rutgers University back in June. Another way that we can detect spotted lanternfly is using something called eDNA, or environmental DNA. And the idea here is that as an organism is moving through its environment, it's shedding DNA on its host, such as Tree of Heaven, and that even if we're not detecting spotted lanternfly using the aerial attractants or using the sticky bands, it might actually be there. And by sampling for DNA on the trees, which the picture here, these individuals are using paint rollers and they're rolling solution on the tree to try to collect DNA from it. We might be able to detect populations at incredibly low densities before they establish. And lastly, our goal is to do outreach. So the invasive insect seminar series, we're hoping to put together pre and post-season reports for different stakeholders. And again, if you have Tree of Heaven present on your property or at your work, and you're interested in participating in this study, please contact me, JC Andersen, that's SEN, at umass.edu. And looking forward to your questions after.
Great, thank you so much, Jeremy. And I will try to swap us over here to our last presenter, Dr. Pinero. Give him an invitation to take over. And we've just heard from Dr. Pinero this morning, so he needs no introduction, but again, he will discuss the risk posed by spotted lanternfly to the mass fruit industry and his part in this project. So take it away, Jaime.
Thank you, Tony. So I only have 6 slides to show, so I'm going to spend the last 5 minutes of this group presentation with an overview of the risks posed by spotted lanternfly to the fruit industry in Massachusetts and some monitoring efforts. So research has been done around the country, in particular in Pennsylvania, and the goal has been to evaluate which crops are at risk. So feeding has been observed in some specialty crops such as apple and stone fruits, but to date, grapevine is the only agricultural commodity on which feeding has been observed directly, causing some damage. So in grapes, feeding can delay fruit ripening and can also reduce the winter tolerance of those plants to the cold weather. In addition, experiments in Penn State University, there was some evidence that feeding on peach trees, on the branches or on the trunk, when the trees are small, can lead to some fruit abortion. So something I want to emphasize here is that actually a spotted lanternfly will not feed on the fruits themselves. They will feed on the trunk, they will feed on branches, they will be sucking up all this sap. So what you see in the picture now is a spotted lanternfly feeding on apple branches, but they are not feeding on the fruit. These other pictures highlight the secondary problem, which is basically honeydew on grape foliage. Of course, I'm including a picture on oak, and the growth of sooty mold. In addition, as you know, it was discussed before, the honeydew can also attract some insects such as wasps, which once are present, some of them may be also feeding on fermenting fruit. So this is a problem for growers, of course. What I have here is a short video where you will see a spotted lanternfly adults feeding on grapevine arms and the cordons and the dripping of honeydew. So we try to point to those honeydew secretions. So again, they're feeding on the sap, but they are not piercing the fruit. Somewhere in the center, you can see on the right at the bottom, the honeydew already deposited on the fruit. And somewhere at least in some seconds, you will see some dripping of honeydew by the flies themselves. Let's see what it is. It should happen somewhere here. So that's the honeydew dripping, and that's basically the aggregations of spotted lanternfly feeding, which can affect the tree health. So I will stop this, go to the next, just to discuss the plans for monitoring. So detection is being led by other agencies, such as MDAR and APHIS. APHIS is an acronym for the Animal and Plant Health Inspection Service of the USDA. So we're going to be evaluating traps, which are called circle trunk traps, which are wrapped around the trunk of the trees. And we're going to be using these traps in combination with a plant volatile, which basically is winter green oil. At this moment, is one of the attractants that is showing some promise. And while new attractants are being developed, we're going to be using wintergreen oil, and the chemical name is methyl salicylate. So then we're going to try to find tree of heaven, black walnut in 10 to 12 locations around Massachusetts. If we cannot find these trees, then we're going to try to find maple or birch trees, so that we can have these traps trying to detect the presence of this invasive pest. Traps will be inspected once every 2 weeks from early May to late September. So these are the 10 locations in Massachusetts. I want to try to include one more in the Northeast. And again, monitoring will start in May, and we're going to check the traps frequently until late September. At the same time, we're going to be disseminating information, research-based information, to growers and other stakeholders in the state. So then if you live in some of those areas where you can see the mark, then please let us know if you have tree of heaven, because that will be the best tree that we can use for monitoring. And of course, we hope not to find the lanternfly in Massachusetts, but it's getting very close. So again, it was just a very quick overview of the type of damage that you can expect, especially in grape, and then just the monitoring efforts that we're going to be doing this year, supplementing efforts by other agencies. So thank you very much. And that's all, Tony.
Excellent, thank you. Well, now's the part where I can open up things to all of our presenters here for some questions from the audience. So I think what I'll do is moderate those questions as I have in the past, and invite our presenters, anybody who feels so inclined, to answer the question. So our first one for the group is from Jan. Is spotted lanternfly more attracted to larger trees than smaller or potted trees that nurseries would ship? So do we have anyone?
I can answer that.
Thank you.
Yeah, the tree circumference or diameter at breast height is actually a very strong predictor of density. So we do find that they prefer larger trees. That's for feeding, all the nymphal stages and adults. But when it comes to egg laying, I'm not sure that tree diameter is, I don't know if it is or not important. They will lay eggs on inanimate objects. So that's the biggest issue, I think, for nurseries is, well, that's one of the issues for nurseries, is to avoid moving egg masses.
Thank you. Yes, I would think that it would be important to check your pots as well as that nursery stock, even if it's small. I know I've seen pictures of small diameter hemlock nursery stock with egg masses laid on them. So definitely something to look out for. Next question for the group from Sean. Why is the tree of heaven the most preferred by the spotted lanternfly?
Again, I think I'll take that one.
Thank you.
So, well, it's a very mysterious insect. We still don't know a lot about it, but we do see that their population really builds up in ailanthus prior to mating, and then after mating, it starts to go down again. So in one study that we did in my group, we did transects where we trapped on trees along transects with different species of trees, and 97% of the spotted lanternflies that we caught just prior to mating were on tree of heaven. So it's my opinion that they're using tree of heaven to aggregate and congregate prior to mating, and it's probably important in mate finding. That's not to say that they can't find their mates on other trees. They do, and we've seen them mating without tree of heaven, but it seems that that's one of the reasons.
May I add something, Miriam? This is Jaime.
Yes.
I would like to add that in China, the tree of heaven is also one of the preferred host plants for the brown marmorated stinkbug. Well, also here in the US. But this tree, for some reason, I don't know exactly why, it has a number of insects that specialize on this tree. There is some weevils, 2 different species of weevils from China. I mean, they're not present in the US, and they only feed on the tree of heaven. So I don't know what is so special about the tree of heaven, but it's a plant that attracts, or of the invasive species, is a preferred host plant for also the brown marmorated stinkbug. Thank you.
Thank you both. I do want to also point out, I've just shared in the chat a resource from our friends at the Mass Department of Agricultural Resources. These are some best management practices for our nurseries here in the state. So again, if you're concerned about potentially moving spotted lanternfly accidentally, you want to definitely check out that document. All right, for the group, the next question from Gary: would spraying the egg cases on the tree with 70% alcohol kill the eggs? And I'll volunteer to hop in on this, and if others have stuff to add, they can. There have been some studies on insecticide options for managing spotted lanternfly coming out of Penn State and other researchers. I can mention that Penn State reports that the most effective ovicidal or egg-killing insecticide that was tested on ornamentals was a product called JMS stylet oil, which is a refined mineral oil-based product. And they report up to 71% mortality of the egg masses when used at 3% rate. And they also note that the control mortality in these studies was 35%, so that they were seeing some eggs not making it even without application of this mineral oil-based product. So that's the information that I have, I think, for egg mass management, and I will share, once I get a minute in the chat, some fact sheets from Penn State about what they've developed for management of the spotted lanternfly. I'll also note that I have not shared management information yet on our extension webpage for this insect because, again, here in Massachusetts, not having an established population, our main focus right now is to report any suspicious life stages of this insect. We're not in the point where we are managing for it. We are looking for it and wanting to prevent it from entering Massachusetts. Does anyone want to add to that at all, or should I go on to the next question? Okay, not hearing anybody jumping to that. So a question from Jan: do the adult spotted lanternfly stay on or near the trees where the eggs were laid, or can they move away from, I guess, where they hatch? Or
again, I can talk about that a little. They do move quite a bit. So as soon as they emerge as nymphs, they are tasting and moving around and trying out all sorts of different plants. So they're very opportunistic. And as I mentioned before, we do see like this increased population accumulation on Ailanthus from early instars to adults. So they start off very broad, tasting all sorts of different plants, but they move around, and then gradually you see more and more of the population ending up on Ailanthus. But they do move around and they don't stay near their eggs.
Thank you. Let's see the next question. We've got maybe time for one or two more. From Gary: do the spotted lanternfly lay their eggs at the base of the trees, or could they be found at any height?
Again, I can address that one. Some folks have in Penn State have been working on that, and it looks like majority of egg masses are above 2 meters, so above where we would be looking. But they do lay them all over the tree.